Hey there, my name is Michael. I'm a fourth year PhD candidate in the Sharpton Lab in the Department of Microbiology at Oregon State University, and today I will be telling you a little bit about my research investigating how environmental factors impact gut microbiome stability to influence host health using the zebrafish model organism. My research explores three key topics of ecology, sensitivity, resilience, and stability. In terms of the gut microbiome, sensitivity is defined as a microbial community's inability to resist change from a disturbance. And that could be represented by the green and red lines here. So imagine we're plotting diversity on the y-axis. So high diversity, low diversity, and then time is on the x-axis. And we have three different microbiome communities as indicated by the three different colors here. A sensitive microbiome would be one where if we disturbed these microbiome communities on day 0 using antibiotics or some kind of parasite exposure, temperature change, for example, the sensitive microbiome communities would experience a great change as shown by the dip in diversity here. Resilience is defined as how quickly a microbial community can return to its pre-disturbance state. So we had the disturbance on day 0. The microbiome communities that were sensitive dipped in their diversity, but then the green line appears to have recovered by increasing its diversity back to a similar pre-disturbance state. Okay, lastly, we have stability, which is a bit trickier to define. And so my research is trying to come up with better definitions of stability in terms of gut microbiomes so that they are quantitative and they're applicable across different host microbiome systems. At this point, it's pretty well understood that gut microbiomes provide many beneficial services to their hosts through a variety of mechanisms. And absent any major perturbations or disturbances to the gut microbiome, they're relatively stable and able to conduct their functions to help the host's health. But when they're disturbed sufficiently, they become dysbiotic or linked to states of unhealthy negative health outcomes. But we don't quite understand the underlying mechanisms that push or pull on a microbiome stability. And so that's where my research comes in. To address this knowledge gap of what factors drive microbiome stability to impact host homeostasis and health, my research is attempting to answer these two questions. So the first question asks, what measurable properties of microbiomes predict resistance and resiliency to a variety of environmental stressors? And I hypothesize that microbiome taxonomic diversity provides resistance and resilience to host microbiomes. Furthermore, the microbiome's functional redundancy improves community resistance and resiliency. And finally, the strength and degree of the interactions among microbiota mediates community sensitivity. The second question asks, how do disturbances and the interactions among them affect the nature of the host microbiome relationship? I hypothesize that disturbances affect the microbiome prior to an observed effect on the host. Moreover, disturbances shift the degree of interactions between hosts and their microbiota and enrich for antagonistic interactions between them. To answer my questions and test my hypotheses, I conducted a longitudinal experiment where I exposed zebrafish to multiple and combined environmental stressors to see how they impacted the zebrafish gut microbiomes to influence their health. So I know this diagram has a lot going on with it, but simply I'm trying to understand how individual and pairwise combinations of these exposures of antibiotics, high temperatures, and or a parasitic worm contribute to impacting the zebrafish gut microbiome stability and how that in turn influences the zebrafish's health outcomes. The results of my study has the potential to identify universal properties that underlie microbiome resistance and resilience that contribute to gut microbiome stability and help maintain host health and homeostasis. And from that, we can use these results to develop system-agnostic microbiome measures to translate across different microbiome host systems. For example, we can identify features that relate to gut microbiome stability in zebrafish, but also in humans, mice, coral, seagrass, etc. And using those, we can diagnose or predict gut microbiome stability in the face of disturbances, and then we can also develop microbiome targeted interventions to maintain or recover gut microbiome stability. And lastly, we can use these to inform management practices and conservation efforts to minimize anthropogenic impacts such as climate change.